Hey everyone, welcome back to my channel. And today I'm going to talk about DNA replication. Specifically, this is a part 1 of video where I'll be talking about the initial process of DNA replication. So if you haven't watched already, I request all of you to watch my previous video on DNA structure as well as a video on nucleic acid, nucleosides and nucleotides, and that will help you understand this lesson very clearly. So DNA replication is a process by which DNA makes a copy of itself. Basically, it's a biological process where two identical DNA molecules are formed from one DNA molecule. And it is important in accurate transformation of genetic information from a parent to an offspring, and therefore it is an essential part of genetic inheritance. Now, it is also essential for cell division during growth and repair of damaged tissue, as well as when new cells are formed, it provides these new cells with their copy of DNA in order for the cells to function. And if any error occurs in the process of DNA replication, that can lead to mutations or changes in the genome, which are associated with several clinical disorders. So there are some of the important things we need to know about DNA replication. First, replication is bidirectional. What I mean by that is, if you know the DNA strand, they run anti-parallel. So these are our two original strand, one in red and one in blue. So they run anti-parallel. So when new strands are synthesized, the new strands are here in green color. So they will also be synthesized in opposite direction. That's why the replication is bidirectional. And just to mention here, this original strand, they are called as parent strand, and the new strand which are being formed, they are called as daughter strands. Second, replication is semi-conservative because each of the parent strand are intact in newly synthesized DNA molecules. So if you can see here, the one in blue and one in red, they are intact in newly synthesized DNA molecules. So that's why they are called as semi-conservative replication. And third, new strands only synthesize in 5' to 3' direction, and you will see this shortly. And lastly, it is also important to know that DNA replication occurs in S phase of the cell cycle. So there are basically 3 stages of DNA replication: initiation, elongation, and termination. So let's first talk about the initiation process. So let's say we have a DNA strands here, and in order for replication to initiate, these two strands must be separated over some area in this DNA strand, and that area where these two strands are going to be separated, it is called origin of replication. So suppose this is our site of origin of replication. Now the question is, why not this area or this area is being called as origin of replication? Now there is a really good reason behind that this site of origin of replication is almost exclusively composed of adenine and thymine. Now the question is, why only adenine and thymine? Why not guanine and cytosine? So if you remember from my previous video, adenine and thymine nitrogenous bases are joined together by 2 hydrogen bonds, opposed to the guanine and cytosine, which are joined together by 3 hydrogen bonds. So the separation of these strands are energy consuming process. So it requires less energy, or it is easy to break 2 hydrogen bonds between adenine and thymine than to break 3 hydrogen bonds between guanine and cytosine. So that is actually the reason behind adenine and thymine is being chosen as site of origin of replication. Now, how are these strands are being separated? So there is a protein which is called pre-replication protein complex. So this is actually called pre-replication. It is easy to remember because it's called pre-replication because it is the process before replication, and protein complex. It's called complex because there are more than one protein which are involved in this process, but I'm definitely not going to talk about those proteins. But you need to remember that there is a protein called pre-replication protein complex which actually comes and bind to this site of origin of replication, and what it will do is it will actually hydrolyzes the ATP to melt the hydrogen bonds between adenine and thymine to initiate the separation of these two strands. So when it does that, when these two strands are being separated over this local region, it creates something which is similar to bubble, and that is called replication bubble. And these two ends of replication bubble, which kind of looks like a V-shaped structure, they are called replication fork. So they are nothing but just the two expanding ends of a replication bubble. So now the important thing is, for DNA replication to initiate, these two strands must be separated further from this replication fork, and there is an important enzyme which play a role to separate these two DNA strands, and that enzyme is called Helicase, which actually unwind this DNA helix by disrupting hydrogen bonds. Suppose we have this helicase enzyme here, it won't unwind these two complementary strands, and now these two strands are separated. However, there is a problem. These two strands are very vulnerable to reannealing or reconnecting to each other, and to prevent this reannealing or reconnection, there is a protein which is called single-stranded DNA binding protein. So this protein will come and it will bind with the single strand with single strand of DNA. So SSBP, which binds with this single strand of DNA, and that binding of this SSBP to single strand DNA will prevent reannealing or reconnecting to each other. Now there is also one important function of this SSBP protein. It actually also protects these two single strands from nucleases enzyme. Now nucleases, what they do is they actually degrade these DNA strands, and they do it by breaking this phosphodiester bond. So if you don't actually protect this strand from nucleases, it will keep breaking this phosphodiester bond that will result into DNA fragmentation, and eventually that can cause cell death. So single-stranded binding protein, not only it keep these two strands separated, but it also protect this strand from nucleases, which degrades these strands. So while helicase is unwinding these strands, there is another problem which occurs. So consider these two strands as a string. So when you pull these two strings apart from each other, it is going to create a torsional strain between these two strings. And what it will do is it will keep these two strings will keep twisting with each other, and eventually it will over twist. And this over twisting of this two strings will somehow look like this, which is called DNA supercoiling. So if there is this DNA supercoiling, it will become very difficult for helicase enzyme to separate these two strands. So to prevent this supercoiling, there is an enzyme called Topoisomerase, so which relieves this torsional strain which is resulted from helicase induced unwinding. So topoisomerase comes in, bind with this DNA molecule just ahead of the replication fork. Now topoisomerase has two important activity. First is nuclease activity. As I mentioned before, nucleases they break phosphodiester bond between two nucleosides. So topoisomerase, performing its nuclease activity, breaks the phosphodiester bond between two nucleotides, which will help relieving this supercoiling between two strands. However, the only problem is if these strands are not resealed, it will inhibit the DNA replication process. And if it keeps breaking this phosphodiester bond, it will eventually result into DNA fragmentation and cell death. So its second activity is ligase activity, that means sealing activity. So the phosphodiester bond which was broken by its nuclease activity, that will be resealed by performing the ligase activity. So the ligase, so that means with its ligase activity, it will reseal this phosphodiester bond in order for DNA replication to continue. Now there are two types of topoisomerase. Topoisomerase 1, which does not require ATP, whereas topoisomerase 2, which is an energy consuming process. Now topoisomerase 2 is also called as DNA gyrase in bacteria. So I also want to mention something else which is very important, that how topoisomerase have been targeted in clinic to treat some disorders. So there is a drug called etoposide which inhibits topoisomerase 2. Now etoposide is used in as a chemotherapy medication to treat various types of cancer. So it is very interesting to learn how etoposide actually inhibit topoisomerase 2. So what it will do is it will actually enhances or increases the nuclease activity. That means it will keep breaking the phosphodiester bond between two. Simultaneously, it will inhibit the ligase activity. So that means it is breaking the phosphodiester bond, but it will inhibit the resealing of this phosphodiester bond. So as a result, what will happen, that it will create this DNA fragmentation and eventually cell death, and eventually this cancer cell won't be able to replicate. So it's very interesting to know how etoposide inhibits topoisomerase 2. Now there is another class of drug which are called fluoroquinolone antibiotic. So if you have heard of ciprofloxacin, levofloxacin, mofloxacin, which inhibit this DNA gyrase enzyme in bacteria, and this has been approved to treat various forms of bacterial infections. So it also works with the same mechanism as etoposide. So lastly, let's just have a quick recap of DNA replication, the initial process of DNA replication. So DNA replication is initiated by forming the origin of replication, where and this origin of replication, which is enriched in adenine and thymine, and that's where the pre-replication protein complex comes in and binds, and which will eventually form a replication bubble, and the ends of the replication bubble forms the replication fork. That's where the helicase enzyme comes in and start unwinding these two strands of DNA, and while doing so, it creates this torsional strain between these two DNA strand that could result into supercoiling, and which can be prevented by topoisomerase enzyme. And lastly, these two separated strands, they have a high affinity for each other and they can reconnect or rejoin, and to prevent this reconnection, there is a single-stranded binding protein which binds to the single strand of DNA and keeps them apart, keeps them separated, as well as it will also protect from nucleases, which are responsible for degrading this single-stranded DNA strands. So this is the basic process of initiation of DNA replication. So in the next lesson, we will be learning about how the new strands or these daughter strands are being synthesized. So thank you so much for watching this video, and if you like this video and if you think you learned something from this video, please like and share the video and subscribe the channel. Thanks so much and have a great day.